So now we continue in the discussion of the book Molecular Immunity, a chronology of 60 years of discovery, and we're on to chapter 4, and actually also going to do chapter 5 today. Chapter 4 is entitled The Molecular Mechanisms of T-cell cytolysis, or killing. And the thing about both of these chapters is that in the 1960s was the beginning of cellular immunology, and there were 3 different cellular immunology assays described in the 1960s that then became the bedrock of the whole field for the next 20 years, 25 years essentially. The first we've already discussed, and that was Peter Knowles' discovery in 1960 that you could add things to cultures of lymphocytes and it would stimulate them to proliferate. So the proliferation assay became really the first assay, and also probably the most important from the standpoint of activating the immune system, the immune response in the test tube anyway. And it was measured by simply adding radioisotope labeled molecules that were incorporated into newly synthesized DNA. So we weren't really measuring proliferation per se, but we were measuring the replication of the DNA in preparation for then the mitosis that occurred a little bit later. So the question that this assay brought up right away was, and when we then, when it was discovered in 1965 that there were these things in the medium of activated lymphocytes that when placed on other cells would make them proliferate, and that became then T-cell growth factor and then interleukin-2, and I'll get into a little bit of that. Once we knew that there were these mitogenic factors that were in the supernatants and in the culture media, the question became then, well, what's the difference between activating the T-cell receptor, which then became known, and we discussed that in the last video. What's the difference between the T-cell receptor and the IL-2 receptor in terms of the signals generated that made the cells behave differently? And that was, in this instance, proliferation. So that was one assay that needed to be sorted out, and I'll describe then how that happened. The next assay, though, was reported only in 1963, which was an assay for the generation of antibody production, antibody forming cells. And this assay was described by a fellow by the name of Niels Jerne, who was a Danish physicist who decided that physics was probably a little too hard for him, so he decided to become a biologist. If I had been around, I would have explained to him that physics is a hell of a lot easier and simpler than biology is. But anyway, so he described an assay where you would immunize mice with sheep red blood cells, and you'd wait a week or 2 or 3, and then you'd kill the mice and take the spleen cells out of those mice and make single cell suspensions and put them into culture in petri dishes that had a layer of sheep red blood cells. And this was done in soft agar, but the agar was warmed to enough so that it was liquid when you put the cells and the agar together. And then you would add to this mixture guinea pig serum as a source of complement. Complement had been discovered, and I didn't go into that in the early part of the 20th century as something that was necessary, and that's why they called it. But they didn't know exactly what it was. It complemented antibodies because it allowed the antibodies to actually cause lysis of the red blood cells that were in the agar. And when you looked at the petri dish and looked down the microscope at the petri dish, he showed that there was only one cell in the middle of a ring of lysed red blood cells that formed a plaque, and that word, the plaque, came from microbiology and virology, where an infected cell would form a plaque that you could see with the naked eye. And that was really the first demonstration that McFarland-Burnett's hypothesis that there was a clonal selection and that only one cell would make one antibody, and that's what he said in his clonal selection theory in 1957. Yerny basically demonstrated that that was true, which was a good thing. And then over the course of the next decade or more, people sorted out the fact that so you needed B cells that would ultimately become the antibody forming cells, and then you needed T cells to give some help. And that's where the phenomenon of T cell help arose. But you also needed macrophages. So this sort of complicated the whole deal, because then the experimental problems was you had to sort all this business out. Now, in addition to that, to the problem where we had all these three different kinds of cells and so forth, it was shown that there were the same kind of medium that had conditioned medium that had T cell growth factor in it, also had what they called T cell replacing factor. You could take the T cells out altogether, but you had to put in the conditioned medium, the T cell replacing medium. And the same thing then was shown for macrophages. So that you could get rid of the macrophages, but you needed culture supernatants from activated macrophages. So that was that was sort of a problem, because you had mixtures of three different kinds of cells. You had soluble things and you had other kinds of things. And so that set the stage then for the 1970s, essentially. Now, the other thing that happened in the 60s was one more assay that had been described, and that was described in 1968, and that was the assay for killer T cells. And that assay was simply what you were able to do at the time was, it was described that you could culture your target cells, usually used a tumor cell, and with a radioisotope. And it was what we used was Chromium 51. So the cells would take up the Chromium 51 and it would bind to different proteins and other molecules in the cell, and it would stay there. And you could then wash off the excess radio label. And then you could take those labeled target cells and mix them together with other cells, T cells and whatever you wanted to mix them together with. And you could then wait for a while. And ultimately, if a cell was killed, the membrane of the cell would become leaky, and it would leak out the radio-labeled chrome that was now attached to big molecules, so it shouldn't have leaked out unless you had a major damage to the membrane. And it would leak out into the medium, and then you could then assay for that how much chromium was released, and that became the assay for killer T cells. So then, in the 1970s, there were all kinds of experiments that were reported by multiple different laboratories over the course of the whole decade, trying to figure out what these soluble factors were. A lot of different factors were just about as many as the different investigators were involved. We had a different name for the factors. We had T cell activating factor, we had T cell killer factor, we had T cell stimulating factor, T cell replacing factor, and the problem was that nobody knew what the molecular basis of these factors really activities. They were activities essentially. So we were back to the same stage that the antibody investigators were in the early part of the 20th century, when they had antibody activity that they could measure by either, as I said, lysis of red blood cells or agglutination of their antigen that they could look at under a microscope slide. So they had agglutinating activities and hemolysin activities. Well, the T cell people, a couple of decades later had the same problem. They had activities, but they didn't know what the molecules were. And that became then the experimental problem that everybody was focused on. That was, actually, if I could be a little bit immodest, we broke the ice on that whole deal. And that in that what up until that time, people just had assays that said, well, yes, there was activity in my supernatant, or there was not. It was an all or none kind of a thing. But what we did was we constructed a quantitative assay so that we diluted multiple dilutions of our conditioned medium, and we could show then that the amount of proliferations, what we were looking at, was commensurate with the concentration of the activity in the conditioned medium. And that was critical because it allowed us to compare one batch to another batch on the one hand, and also allowed us to fractionate. We take a batch and we put it through all kinds of biochemical separative procedures, and we test each fraction as it came off the different columns and so forth and so on, and we could then find where there was a peak of activity. And that allowed us essentially then to purify T-cell growth factor to homogeneity. It allowed us to get an antibody that would bind to it and react against it. And that allowed us to get to the molecule and then the gene beyond that. Well, that whole sequence of events had to happen with these other assays. So we did that for T-cell proliferation, but you also had to do that with the antibody forming cells and also with the killing aspect of things as to what was actually going on. And so the first thing that I wanted to focus on was the biochemical events that occurred immediately after activation of the T-cell antigen receptor, the so-called T-cell receptor, versus the IL-2 receptor. And it wasn't until 1979, so we passed all the way through the 70s. In 1979, there was a young woman by the name of Carol Deutsch who was interested in excitable cells because they were neuromuscular kinds of cells. She was an expert in this area, but she applied her assays to lymphocytes. And she found that just like the nerve cells or the muscle cells, there was a membrane potential across the gradient across the membrane, which meant that the lymphocytes were excitable cells, which was an interesting thing. And then in the early 80s, there were aspects of the technology that improved so that they developed fluorescent dyes that would bind on to different ions, calcium being one of the most important for the T-cell compartment. And they found that when they activated the T-cells via the T-cell receptor, that within minutes, we used to have to wait 2 or 3 days for them to begin to synthesize new DNA, and so that we could assay for that. But within minutes, you'd see a huge rise in intracellular calcium. And so that gave the T-cell receptor crowd, they got all excited about this because now they had their own assay really for T-cell activation, and they didn't really care what happened downstream from that. That was irrelevant to them and just secondary or tertiary or quaternary to where the real action was being taken place. So early on, you could block so they found that the calcium was going from the outside of the cell to the inside of the cell. And that was what was causing this rise. And the calcium was going through calcium channels. There were channels that actually opened up in the membrane, and the calcium just started flooding in. And and you see these kinds of things had already been worked out in neurons and also in muscular cells. So the assays themselves were pretty well, established in those other kinds of cells. Now they're just being applied to lymphocytes. Well, it was found early on that if you block the calcium from coming from the outside into the inside, then the cells would not proliferate. But, and this is where I sort of argue with Ellis Reinhardt, to try to tell him that the T-cell receptor is just the beginning, This is that if you added IL-2 on top of that, even though the calcium was blocked on the outside, the cells would still proliferate. So you didn't need the calcium rise in order to for proliferation. You did need it for the activating events, the initial activating events that happen through the Tcell receptor. So that's where things stood around 1980, 81, 82. And it wasn't until 1984 when Ellis Reinherz, because he had generated monoclonal antibodies reacting with the Tcell receptor, and we had antibodies reacting with the IL-2 receptor and so forth, that it could be sorted out that there were different biochemical signaling events coming from the Tcell receptor as compared to the IL-2 receptor. So now I want to shift to the molecular mechanisms that are associated with T-cell killing, T-cell cytolysis. As I said, in 1968, Ted Brunner and Jean-Charles Serratini from Lausanne, Switzerland, first described the chromium release assay. And it was found out early on in experiments done once they had this assay that calcium was also necessary for this thing to happen. The cells would not become cytotoxic if you blocked calcium, or if you already had generated cytotoxic T cells and then chelated took all the calcium away, they couldn't kill. So the calcium was very important for killing. That was 1968 and into the 70s, but it wasn't until 1980, 12 years later, that things started to happen with regard to understanding exactly how the T cells were killing these cells. And in 1980, there was an investigator at the NIH by the name of Pierre Henkart, who took electron microscopic pictures of target cells that had been mixed with T cells and in the presence and absence of antibody and so forth, and showed that these target cells, once they had been started to be damaged and were releasing their chrome, they had big holes in their membranes. It was like somebody came along and just put a pipe through. And they were huge. You could drive a truck through these things. They were so big. And so they had these pictures, these electron micrographs that showed that there were these holes. And so the other thing that was known at the time is that antibodies, together with complement, would make the same kind of holes. They'd look the same under the microscope, under the electron microscope. So that was sort of interesting. T cells could also put holes in membranes, just like antibodies and complement. And so there entered a guy into the whole mix of this business by the name of Eckhard Podack. And Eckhard was an interesting guy. I really liked him. He was originally from Germany, and he trained with Hans Müller Eberhard, who was a complement biochemistry person who was at the Scripps Institute in La Jolla, California, and who purified and characterized all of the complement molecules. So Eckhart really was uniquely, he was at the right place at the right time, essentially. And what he did was he got a hold of our clone T cells, and he collaborated then with another investigator by the name of Gunther Dennart, another German guy who was up in LA at USC, who had made clones out of natural killer cells, which could also, obviously, they could kill. So they grew up huge numbers of these things, a liter of those things, so that they had 1 billion cells of clone T cells and clone natural killer cells. And so that from those, they took electron micrographs of these cells, And they found that they were chock full of what looked like granules. They were what the electron dense granules, because when they had all these granules in the cytoplasm, the electrons couldn't get through there. So making the picture. So they had these electron dense granules, and then they figured that they said, well, you know, maybe those granules are how the cells are killing. And so what Podak did then was that he got together with another investigator in New York City, Zanville Cohen, and they decided to see if they could purify these granules. And, well, actually, Eckhart had done that beforehand. He purified the granules from the cells, broke the cells open, got the granules, and then showed that those granules, if you mix them with red blood cells as target cells, that they would lyse those target cells, just like antibody and complement would do the same thing. So then Podak got together with Zan Cohen in New York, and they took all the granules that they could prepare, that they knew how to do that, broke the granules open, and then purified the molecules inside the granules. And they found there were a whole bunch of different proteins that they could see using their biochemical procedures. And one in particular could be reduced to a 75,000 Dalton protein. A Dalton is the molecular size measurement of atoms. So it's 75,000 Daltons. And he then named that P1 for protein 1. And another investigator, Jörg Schoch from Lausanne as well, did exactly the same thing and found that, and he called it Perforin. And actually the name Perforin was, you know, it was just sort of like T-cell growth factor. It says its name. And so that stuck. Perforated the cell. Then in 1989, so this took a decade. And now Eckhart went on and he identified and cloned the gene that encoded the perforin protein. And now with the sequence of the nucleotides that then codes for the sequence of the amino acids in the protein, and they could reveal that the complement and perforin must have come from an ancestral gene, because they had all kinds of homologies amongst them, and that's how you do that sort of thing. And then at the same time, during the 80s, so what they found is that if you could take the purified protein, perforin, and it would poke a hole in the membrane, and it would cause chromium release. But there was other things that were going on at the time that other people had found, and that was that within an hour of the first detectable chromium release, which happened within an hour, or 30 minutes or so, the DNA of the target cell seemed to sort of begin to disintegrate. And it started to fall apart, but it fell apart in a very regular fashion, so that there were just every 200 base pairs of the DNA that would seem to be cut. And there was a whole ladder of these things that you could show on gels, that there was something funny going on. And other people had shown that if you added into this mixture enzyme or molecules that blocked enzymes that would degrade protein, that you could stop the process or at least slow it down considerably. So there seemed to be some stuff going on inside those cells once the perforin punked the big hole in the membrane, that other things were going on, and the hypothesis that then became that there must be proteolytic enzymes that the killer T cell and the natural killer cell actually squirt into the target cell, and then once those enzymes, proteases get inside, it just digests everything else. sort of like putting it into your stomach, and then everything starts to get chopped up in little pieces. And then in the late 80s, in 1986, Chris Bleakley and Irv Weissman demonstrated that there were these protease molecules inside there, and then Jörg Schoep found 8 different proteases and gave them a new name. It helps to name things in biology, because if you can name things, then you become immortal in the science. And so so we named these things granzymes. They were in the granules, and the zyme part of the word means a molecule that will catalyze other kinds of things. So it's a catalyst, and this catalyzed proteolysis, or the lysis of the protein, killing of the protein. So we had granzymes, he lettered them A through H. And that really then by 1990, the whole mechanism, the molecular mechanism of T-cell cytolysis was pretty much well understood. And so it took from 68 to 79 to go from Chromium release to T-cell clones. And then having the T-cell clones and having them be functional T-cell clones, cytotoxic clones. It took another 10 years to do the basically all the difficult hard work to identify the granules inside, and to find out what was inside the granules, and then to purify it and to do the deal, and there you go. So it took 20 years, but that's one of the reasons that I've labeled the title of my book Molecular Immunity, because this is, once we got down to the molecules, then we knew which things that we might be able to manipulate to the advantage of the physician or so forth.